So in the previous lecture, we focused on how LNPs are engineered, the chemistry of ionizable lipids, helper lipids, cholesterol, and PEG lipids, and how formulation parameters determine the physical properties of the particle. But once an LNP is injected into the bloodstream, the particle we engineered changes because of proteins in the bloodstream. So we will see how these proteins play a central role in determining tissue tropism and delivery efficiency. So once LNP enters the bloodstream within seconds, so many of the proteins in the blood bind to the particle surface. So this layer of adsorbed proteins is called the protein corona. And this actually changes the identity of the nanoparticle. Cells in the body do not interact with the bare lipids that were designed in the lab. They interact with the proteins attached to the particle. So that means the protein corona becomes the biological identity of the particle. So that means, the composition of the corona strongly influences which organs take up the particle. So from a modeling perspective, this creates an important and interesting challenge. We start with the chemical features of the particle, but what actually controls targeting is the corona that forms in the blood. So the most consistent observation of LNP delivery is liver accumulation. And the reason is actually the protein corona. So most ionizable lipids have pKa values around 6.2 to 6.5, which means they are largely neutral at physiological pH. So ApoE in the blood gets adsorbed to these particles. So because of this coating of ApoE, so these particles actually look like a natural lipoprotein. And liver hepatocytes express LDL receptors that recognize these particles, and so the particle gets internalized through receptor mediated endocytosis. For modeling, liver accumulation is a default prediction for any standard neutral surface formulations. So you have to deliberately change the chemistry to direct particles elsewhere. So as we just discussed, and neutral LNPs default to the liver, but organ targeting can be redirected by changing surface charge. And this is done by adding a salt lipid to the formulation. So a negative surface drives uptake, mainly uptake in the spleen, for example. And then we know that near neutral surfaces follow the lipoprotein pathway into the liver. A cationic surface accumulates in the lung, for example. For modeling, the key insight is that the small shifts in zeta potential produce large nonlinear changes in organ distribution. But why does surface charge control targeting? So that comes down to what happens the moment the particle hits the blood, which is what we will see in the next slide. So when nanoparticles enter the bloodstream, many different proteins compete to bind to the particle surface. So there are typically two types of layers are formed. So the soft corona contains loosely bound proteins that exchange rapidly with the surrounding plasma. Beneath that is the hard corona, which consists of proteins that bind more strongly and remain attached to the particle. So the composition of this corona depends on the surface chemistry of the particle. Properties like surface charge, hydrophobicity, and lipid composition determine which proteins attach. So different proteins lead to different biological outcomes. Some proteins promote immune recognition and clearance by macrophages. Some other proteins, for example, reduce immune clearance and help nanoparticles circulate longer. So from a modeling perspective, this means that surface chemistry does not directly determine organ targeting, but what it determines is actually protein corona, which then controls biological interactions. So now that we understand how the protein corona forms, we can identify which particle features actually matter for predictive modeling. So the three most important measurable features are surface charge, particle size, and polydispersity index. Surface charge, measured as zeta potential, is the dominant driver of corona composition and therefore of organ targeting. Size matters because anything above 150 to 200 nanometers is more readily captured by the spleen and macrophages. PDI reflects population uniformity. A high PDI signals aggregation, which accelerates immune clearance. Some features only matter through their interactions. So the pKa of the ionizable lipid doesn't determine charge on its own. It's the combination of pKa and physiological pH that sets the effective charge of the particle in the blood. Similarly, a peg behavior depends on both the density and anchor length together, which jointly control how quickly the peg layer sheds and exposes the particle surface. So these interaction terms are important to include clearly in any model. So when studying RNA delivery, we measure two distinct types of outcomes. So the first is pharmacokinetics. So the question is, where do the particles go? The standard metric is a percent injected dose per gram of tissue. So this is measured across organs, like liver, spleen, lung, kidney, at defined time points. Importantly, these values behave like compositional data. They sum to roughly 100%. So an increase in one organ necessarily reduces availability for others. The second is pharmacodynamics. Did the delivered RNA actually produce protein? So this is typically measured using a reporter signal or direct protein concentration. And this is where the critical distinction we need to know. Expression requires cell uptake, endosomal escape, and ribosomal translation. all of which can fail independently after the particle has already accumulated in the tissue. So high accumulation does not guarantee high expression. For modeling, so this means biodistribution and expression are two separate prediction targets, and a model trained on one will not automatically transfer to the other. And predicting where nanoparticles end up is actually trickier than just predicting one organ at a time, and that shapes how you build the model. So first we have competition problem. That is, organs are pulling from the same pool of circulating particles. If the liver takes up most of the particles, there are simply fewer left for the spleen or the lungs. So you cannot treat each organ as a separate prediction. They are coupled. Second, and again related point is that the dose is finite. The fractions going to each organ have to add up. That makes this compositional data. If your model predicts each organ independently, the numbers won't sum correctly, and you will get outputs that are physically not possible. Third, species matter. The corona that forms in mouse blood is not same as what forms in human blood because the plasma proteins are different. So a model trained on mouse biodistribution data won't necessarily translate to humans. LNP delivery can fail in several distinct ways after injection, and each failure mode has a different mechanistic cause. The first is RES sequestration. Macrophages in the liver and spleen recognize and phagocytose particles, particularly those that are large, aggregated, or have high PDI. So this is, the dominant clearance pathway, this reticular endothelial sequestration, endothelial system, as the dominant clearance pathway for most nanoparticle formulations. The second is rapid systemic clearance driven by PEG loss. So when PEG molecules shed from the surface, the underlying lipid is exposed to plasma proteins. Opsonization follows quickly, and macrophages clear the particle before it reaches its target. A third is off-target accumulation. If surface charge drifts, the protein corona shifts, and particles can end up in the wrong organ. Fourth is immune activation. So highly cationic surfaces directly trigger complement and inflammatory pathways, leading to cytokine release and elimination. So this is also a toxicity concern. Because biodistribution experiments are expensive and require animal subjects, data acquisition needs to be organized. You start with a large formulation library and use in vitro assays to eliminate unstable candidates and anything with poor encapsulation efficiency, for example. Successful ones move to a small in vivo pilot, typically 3 to 5 animals, to rank formulations by biodistribution. Only the most promising candidates then receive full PK/PD characterization with larger cohorts and multiple time points. ML can make this process a bit more efficient through active learning. Rather than selecting the next formulations to test arbitrarily, the model can flag which candidates carry the highest prediction uncertainty. Those are the experiments that would add the most information. So each round updates the model and tightens the next selection. So finally, the training data can be expanded through augmentation, incorporating published biodistribution datasets and using in vitro assay results as auxiliary features alongside the in vivo labels. Okay, so let's close by connecting the full chain from formulation to biological outcome. So lipid composition determines the initial physicochemical properties of the particle. Manufacturing and mixing conditions then set the final particle properties. Once in the bloodstream, the protein corona forms and establishes the identity, which controls both the where particles accumulate and which cells express the delivered payload. And then targeting works through generally two routes: ApoA mediated to liver hepatocytes, and then salt lipids to shift surface charge. Anionic charge redirects to spleen, cationic charge redirects to lung. So basically, LNP design is a multi-objective optimization. Potency, tropism, and toxicity are coupled. A formulation change that improves delivery efficiency often shifts tissue targeting or increases immune activation. So that coupling is exactly what makes computational modeling valuable in this space.